speaker is Amanda Jack, also from Denison University, and she'll be speaking about robustness of structure-based dynamics in molecular motors.
I'm Amanda Jack, and I've been working in Dr. Tugger's lab at Denison University. And this summer I've been studying the robustness of structure-based dynamics in molecular motors. So as Ian said, molecular motors are proteins, and they transport cargo within a cell. So you can see here, this is an example of kinesin, and it is walking across a microtubule with this two-head hand-over-hand motion, as Ian mentioned, and it's dragging this vacuole. And so they're obviously vital to cellular function. Without them, we could not survive. The ones that we study in our lab especially are myosins. Some other examples are kinesins here and dynein. And as I said, they walk along actin filaments, microtubules, to perform functions. We can study them in several different ways. One is through X-ray crystallography, and that looks at it's an experimental method that looks at the structures and tries to identify where the amino acids are within the protein. There are also molecular dynamic simulations that we do a lot in our lab, and that takes all the amino acids and subjects them to some sort of force field within a computer simulation and looks at how their positions change and adapt over time. And then there's also normal mode analysis, which I will be talking about in a minute. The reason that we study these proteins is that their dynamics allows us to predict their function. So by looking at their dynamics and how they change over time, we can learn more about the binding regions, where they bind to actin or where they bind to cargo. We can learn about how they walk, the forces involved in their walking mechanisms, and the conformational changes that are necessary for them to bind and for them to perform their cellular function. So here we have an example of myosin 6 and its motion throughout the cell. So the question that I've been looking at are how robust are these dynamic simulations that we run? We get our structures from the Protein Data Bank, which is an online resource that has X-ray crystallography created structures of proteins. However, a lot of times these structures are not fully resolved because these proteins are highly mobile and often along the surface regions. As you see here, this is 189 myosin 5. You can see that these highly mobile loops are not able to be resolved by protein or by X-ray crystallography. And so we're not really sure where these amino acids are in the protein. For instance, 189, this protein here, is missing about 10% of the protein. And so the question is, if we're basing our study of dynamics on the structures and then using our dynamic studies to determine their function, how accurate are these dynamic studies? And can we study the dynamics of these incomplete proteins? So the method that I've been looking at to understand these partially unresolved proteins is normal mode analysis. And it's one of the simplest dynamic simulations. Basically, it takes this protein and uses a contact map to model this protein as a system of nodes and springs. And then does a lot of math to find normal modes of the protein, which are just basically the most basic motions that the protein can take and how the protein is most likely to move within its function. And a lot of times what you find is that the lowest frequency modes describe the conformational changes that we see experimentally. So by using these simulations, we're able to predict the dynamics of the protein. And NMA is advantageous for several different reasons. It's a lot faster than molecular dynamics because it's such a simple model, and so then our simulations are a lot faster to run. It doesn't require a very high resolution protein. You can use a very coarse-grained model and it's still robust. And it's also incredibly robust in general. It tolerates sequence mutations pretty well, and as I said, the lowest frequency modes often match the experimental data. So then the question is, when you have these partially unresolved proteins, is NMA still robust? An example of NMA robustness here, you can see in this protein, these are the experimentally determined open and closed structures. And then on this side, you have in red the experimentally determined open structure, and then the closed structure is predicted by NMA. And you can see that these are very similar structures. So what I did to look at this and the robustness of NMA, I took 5 molecular motors, 3 myosins, a kinesin, and a dynein, and I removed different sections of the protein. I tried to take sections of different lengths, and I also looked at different regions. For instance, the surface regions that you see on the outsides here, some from the inside, and then also took some out from the hinge regions, which is basically where the protein is going to change when it changes conformation. I also looked at different kinds of structures of protein. For instance, alpha helices, you can see an example here, beta sheets, as you can see up here, and then unstructured regions in that area. And I ran NMA simulations on those. So when I was looking at the data that NMA was producing, the values that I was particularly interested in were B factors, which are a measure of mobility of each amino acid and how much it's likely to move within the protein. And I would I would compare the original model B factors with the model that was missing certain sections. I also looked at displacement of the amino acids within the protein. And I used Spearman correlation, saying that the B factor correlation greater than 0.92 was acceptably close between the edited model and the original model. You can see examples of these B factor graphs here. This is an example of a 0.95 correlation. The green is the original model, and the black is with about 0.2% removed. And you can see that these match pretty closely. There is some variation here, but overall, it seems like a pretty decent model. Here, it's about 20% removed, and that produced a correlation of 0.91. Again, the green is the original model, and you can see that that is significantly different. And then I just tried to determine which types of protein and which areas of protein were the most robust. So you can see an example of the graphs here that present removed on the x-axis and the correlation on the y-axis. You can see a pretty clear negative correlation. As you remove more of the protein, the correlation decreases, which is expected. And one of the largest removal sites that still had an accurate correlation was on myosin 5, only 9. Removed about 21% of the protein, and it produced a 0.93 correlation. So you can imagine that this is a pretty significant chunk of the protein that's removed, and NMA is still behaving remarkably well. So just to summarize the results, surface residues, you could remove about 15% of the protein before the NMA started to break down. The hinge and inside residues, you can remove fewer of those. There was some difference between the alpha helices, beta sheets, and unstructured regions. However, that difference was less significant, but there was a clear distinction within where you're going to remove these proteins. This is an example of a model of one of the partially removed structures. This is about 20. The dark blue region represents the protein that was not present in the model that was removed, and that was about 27% of the protein. So in conclusion, it would appear that normal mode analysis is highly robust when you take limited numbers of amino acids out of the protein. And it's also especially robust in surface loops, which is good news for us because usually the protein structures that we get from PDB are the unresolved amino acids are going to be in the surface because those are the most mobile regions. So generally we're justified in using these incomplete structures in our dynamics analysis. However, this did provide some criteria for the use of partially unresolved proteins, and we can look at that in the future in order to determine whether a structure is reasonable to use in our analysis. Thank you. Are there any questions?
Questions for Amanda?
So you said it's computationally a lot faster. Mm-hmm. How does the accuracy compare to other models?
It's actually extremely accurate. There have been a lot of studies done of NMA. I don't have any data to show here, but it has in the past really accurately predicted the conformational changes that are present in proteins.
It's a related question. So do you have, is it spectroscopic data that you compare against the predictions? I mean, you showed in your Spearman correlation plot, you showed what superficially anyway looked like spectra. Yeah, those, so,
The B factors on the y-axis here.
yeah, I guess what I'm wondering is, what is the experimental verification? What's the, what kind of tools do you have for comparing model with data?
Because I was making my own models, these are not really experimentally correlated. I'm not sure if that answers your question.
Well, we could talk about it some more. Okay, thanks.
Any other questions?
The models that you were working with, you mentioned that some of the protein models weren't fully resolved, but were all the models that you were working with fully resolved?
No. See, I was taking, for instance, 109 is about 10% missing, and then I was using that as the original model,
And you were further taking?
and then further taking off to try to see if I had if I had a complete 109 structure, would removing 10% of the protein make a difference in the structure that I have now that I'm trying to work with?
Okay, let's thank Amanda